This is online lecture 14 for Bio 240, and in this lecture we're going to be talking about translocations. In the last lecture we started talking about chromosomal rearrangements, which were large-scale mutations on chromosomes, including things like duplications, deletions, inversions, and translocations. So the translocations are what we're going to focus on today. And a translocation is a chromosomal rearrangement in which part of the chromosome moves to a different chromosome. So the main point about translocations is that you have movement across trans from one chromosome to another chromosome. And there are two types of translocations. There are non-reciprocal translocations. So this would be, let's say we have two chromosomes. Call this 5 and this one 17. And you might get a break, like we talked about DNA breaks last time. So let's say you get a break right here in chromosome 5. Well, we've talked about you could reinsert itself, it could invert itself back onto chromosome 5 there, or the third possibility is it could attach itself onto a different chromosome. And that would end up with a short chromosome 5 and a normal chromosome 17 that has a little extra bit on its end. So that's a non-reciprocal translocation because one part of the chromosome moved to another, but it didn't get anything in return, as opposed to reciprocal translocation. So if we were looking again at our two chromosomes, here's 5 and 17. And it happens sometimes where there will be a break in 5 and there might be a break in 17. And if these chromosomes are close to each other when these breaks occur, it could be that when they rejoin, they join onto the wrong chromosome. So you'll end up with chromosome 5 that has a little bit of 17 on it, and you'll end up with a chromosome 17 that has a little bit of 5 on it. And you can call these, I like to call it 17 prime or 5 prime, and that's to show that it's the mutant chromosome, one that has undergone a translocation. So you can see here in a reciprocal translocation, the two fragments where there were DNA breaks have now swapped locations. So what are the consequences of translocations? Well, as we saw with the inversions, if the DNA break happens to be at the point of a gene, you can have loss of gene function wherever that break is. Also, like we saw with the inversions, you can have position effects. So now that the pieces of chromosome are in different locations, actually not even on the same chromosome, so the regulatory regions can be altered, and the genes that are in that translocation piece might now be under different regulation. So this is altered regulation. And remember that that changes how and when some of these genes might be expressed in a cell, which cell types, when during development, when during the day, or different stimuli that might change how a gene is expressed in a cell. And then the third consequence we talked about is altered pairing of homologs during meiosis. So remember we talked about that with inversions, and you get those inversion loops which cause the gametes to have abnormal chromosomes. Well, you get altered pairing with translocations as well, but the exact details of it are a little bit different. So I'm going to work out for you what goes on during meiosis with translocations. And remember that pairing is a problem for heterozygous translocations, which is normally what you'll see. For homozygous translocations, because they're the same on both chromosomes, you wouldn't have any problem with pairing during meiosis. So that's where we're talking about heterozygous state. So let's see what would happen if we have a translocation. Let's look at this. Would be one copy of chromosome 5 would be normal. And we have over here chromosome 17. Let's just say it has these segments, F, G, H, I, J, K. And we'll have a second copy of chromosome 5. Let's say there was a break right here, and there was a break right here, and a translocation happened between 5 and 17. So you end up with a 5 that has I, J, K from chromosome 17 on it, and a 17 that has a D and an E on it. So how would these chromosomes be able to line up during prophase? So we would have, remember, when we enter meiosis, we've undergone DNA replication. So we'll have this would be our chromosome 5, and this will be our 5'. They're going to line up at their centromeres. But this area right here can't pair. And then the same would be true. I'll do with 17. Where they can line up at their centromeres along most of their length, but these regions can't line up, can't pair. And so what you're going to get is what's called a tetrad. And let me draw it out for you here. So I've drawn out what this tetrad would look like, where this is our normal chromosome 5. This one is going to pair along most of its length. That's 5'. But then the part that has the translocation is going to try to pair with another part that has that same homology, which is part of our normal chromosome 17. And then the same thing above, our other chromosome 17 will be able to pair at the part that's homologous with regular 17. This one's 17 prime. Whereas the part that has the translocation is going to pair with the other chromosome 5. So you can see now we've paired up everything with homologous regions, but in order to do that, the chromosomes have had to bend and form this kind of tetrad structure here. So what happens during the rest of meiosis? Well, remember, I'm just going to draw it out on the side here. Remember that in prophase 1, we have our homologous chromosomes lined up along what's called the metaphase plate, and then we have these spindle poles at either end of the cell, which attach with microtubules to the centromeres of homologous chromosomes, and that's going to pull these chromosomes to either pole of the cell, and now you have your place for cell division to occur. So during anaphase, your spindle poles are going to attach to the centromere and pull each homologous chromosome to one pole, one side of the cell, so that it can undergo cytokinesis and form two daughter cells. But if you look at this tetrad, that's going to be a problem. So how might this chromosome tetrad get pulled apart during anaphase? Well, it could be that it's lined up like this, and so you'll get a spindle pole attaching to each of these, and you could get 5 and 5 prime going to one side, and on the other side you could get 17 and 17 prime at the other pole. And if that were the case, now you're going to end up with one daughter cell that has the normal 5, and you're going to end up with, that same daughter cell will also have the 5 prime. That's the translocation part. Whereas the other cell at the end of meiosis 1 is going to have the normal 17 and the translocated 17. So they'll have F, G, H, and then the translocation portion. So that's after meiosis 1, and then remember we go through meiosis 2. We have another round of cell division where the sister chromatids are pulled apart, and so we're going to end up with our 4 daughter cells at the end of meiosis. We're going to have one that has 5 and 5 prime, another one that has 5 and 5 prime. You're going to end up with, from the other one, you're going to have another daughter cell that has 17 and 17 prime. So you can see here that none of these cells is correct, has the correct amount of of each chromosome 17 and chromosome 5 material. For these two cells, you end up with duplications of that region, and then deletion, you only have this much of 17, so all the rest of 17 is deleted. So you have large duplications and large deletions, which is going to result in a non-viable offspring or gamete. And same thing is true on the other side. Here we have duplications of this portion of chromosome 17, and we're missing the majority of chromosome 5. So those gametes will be non-viable. So if the tetrad had arranged itself so that 5 and 5 prime and 17 and 17 prime were pulled to the two opposite poles together, you're going to end up with all non-viable gametes. But there's another possibility. Let me just get the tetrad back. So here I've drawn the tetrad again, but this time I've just shifted it 90 degrees, and you can see, so it could be now that this centromere and this centromere are pulled to one pole, and that would be chromosomes 5 and 17 prime could end up at one pole, and then this one and this one could get pulled to another pole, so we'd end up with 5 prime and 17 as the two daughter cells after meiosis 1, separation of sister chromatids. Let's look at what the gametes would look like here. So why don't you pause and try to draw out what the gametes look like. So this is what you should have gotten. We're going to have two daughter cells that have a normal chromosome 5 and a translocation on chromosome 17. So this is going to result in a duplication of this segment DE and a deletion of the segment IJK. And on the other side, you're going to have a normal copy of chromosome 17, but the translocated copy of chromosome 5, and this will result in a duplication of the IJK region and a deletion of DE region. So again, we have in all four of these, we have large duplications and deletions, resulting in all non-viable gametes. Now there's a third possibility for how the chromosomes can be segregated, and you have to kind of picture it in 3D because I can't really draw it here too well, but it could be that 17 prime and 5 prime go to one spindle pole, and 5 and 17 go to another one if they're arranged just right along the metaphase plate. So you'll end up with 5 and 17 and 5 prime and 17 prime over here. So now take a minute to draw out the gametes from this one and decide which ones are viable and which ones are non-viable. So you'll get two cells that have a normal copy of chromosome 5, a normal copy of chromosome 17, and then two cells that have the translocation of chromosome 5 and the translocation on chromosome 17. But you'll see that in these gametes here that have this combination, you end up with a full complement of segments from chromosome 5 and a full complement of segments from chromosome 17. So they do have all of the genetic material. Now, assuming that whatever gene was disrupted at the breaks here and any of the positional effects didn't affect gamete agenesis, then these gametes would be viable. So we would end up with 4 viable gametes in this situation. So if you have someone with a heterozygotic translocation, only 1 out of 3 possible arrangements during prophase 1 will result in viable gametes. So overall, a person who has this translocation, a heterozygote with this translocation, will have reduced fertility. So I just walked you through all of this, assuming that there is no crossing over. But what do you think would happen if there was crossing over during prophase 1? Would you get more or less viable gametes? I think so. Try that for yourself and see what you come up with, and I will see you in class.